male song production in a species called Drosophila virilis. So at events like these, we come together for the sole purpose of communication or to exchange information with one another. Unilateral communication is exemplified by something like this seminar, where the lecturer provides information to the audience in a one-way direction. However, bilateral communication is exemplified by something like a poster session or a networking lunch, where individuals can exchange and modify information with a partner. Thus far, we have only been able to study the neurogenetic basis of unilateral communication using the genetic model Drosophila melanogaster. In this species, only the males produce an acoustic signal of pulses and hums during the courtship song. You see the courtship going on here. However, it was published nearly 20 years ago from the Hoikala lab in Finland that both males and females of the Drosophila virilis group produce an acoustic cue during the courtship. This would lend us the opportunity to study the mechanisms of bilateral communication. So we confirm the presence of this bilateral communication, and I'll show you that with this video. This is our courtship assay, and this is a plastic beveled chamber with the bottom cut out and replaced by mesh. Underneath of that mesh is a pressure gradient microphone that will pick up small air movements created by vibrations of the wings. And I'll point out here, this species is monomorphic, so this is the female, and this is the male. And hopefully what you're going to see is that both of these individuals will vibrate their wings, producing two distinct types of sounds. You should hear it now. Hopefully you guys can see and hear that. And what I'd also like to point out, what's going to be important in a little bit, is that when the female accepts the male song, she physically spreads her wings in this purposeful motion to let him know that she's receptive and ready to mate. And he cannot mate unless she does this behavior. You'll see and hear this again. And here's the wing spreading. And the successful copulation. So we're interested in the behavior itself and the neurophysiology and the genetics that underlie this bilateral communication. And we're currently working on developing some neurogenetic tools for this species, but today I'm just going to mostly tell you about the behavior. So this is what the song looks like in a oscillogram. These are female pulses and what we call male bouts, or dense stereotyped regions of male pulsing. And we wish to not only call this bilateral communication, but hypothesize that it's actually an acoustic duet. So we define duet as a predictable acoustic interaction, and we wish to satisfy 3 criteria in order to call this a duet. We want to show that both songs are important for the success of the courtship, that the songs are coordinated in time, and that hearing is involved in the patterning of song. So in order to assess the importance of song for the courtship, we looked at latency to mating assays. So these plots that you're going to see are represent percent copulation of multiple pairings over a 30-minute observation window. And so in wild-type conditions, copulation is robust. About 80% of them actually copulate. And in order to remove the acoustic stimulus from the courtship, we remove the fly ear, or the arista, which is a feathery structure on the antenna which transduces air movements created by vibration of the wings to the brain via mechanoreceptors. When the female is deaf, there's a significant reduction in copulation success compared to the control. And this is consistent with other data showing that male song is required for a successful copulation. However, when the male is deaf, there is no difference between the experimental condition and the control. So this would suggest that the male actually doesn't care about female song. But one confound of this manipulation is that the male can see that wing spreading that the female does. So in order to eliminate this visual cue for receptivity and assess only the song, we put these deaf males in the dark. And in this condition, copulation success is reduced by half. So female song is important for mating success, but only in the absence of this visual cue. In addition, we've shown through a remating assay that females, receptive females, sing more than those that do not remate, which might also indicate that the female song is a receptivity cue. The Stern Lab Forum, along with our lab, has developed a multi-microphone system to record 32 courting pairs simultaneously. Because we can easily distinguish the sex origin of the song, whether it's male or female, on the basis of the pulse frequency profile and the time between pulses or interpulse interval, We developed a MATLAB-based code in order to automate the song segmentation. It detects male bouts in blue, female pulses in red, and potential regions of overlap in green. So with the efficiency of this automation, we can score hundreds of flies very easily and begin to answer our question about whether or not the songs are coordinated in time. So we first analyzed female response time, or the time between the beginning of a male bout and the following female pulse. The data are going to be plotted here as normalized histograms. So in red, what you see is the distribution of about 1,000 female response times. In blue is the representative location of a male bout, and in gray is a randomized version of our data set. So hopefully what you can see is that the female response is significantly different than random, and it's peaked at about 410 milliseconds from the onset of the male bout. Likewise, we analyze male response time, or the time between the beginning of a male bout and the previous female pulse. The response times are once again significantly different than random and peaked at about 110 milliseconds. Therefore, this shows that both males and females are coordinating their song with their partner in a non-random fashion. So to assess the third part, if hearing is involved, we once again return to the deaf manipulation, the removal of the hearing organ. So this is a 20-minute representation of a wild-type song where both the male and the female sing. And when we remove the arista from the female, thereby deafening her, she upregulates her song production tremendously. And so this rate is quantifiable and significantly different than wild-type. And this suggests that the male acoustic cues actually suppress female singing in the wild-type condition. Next, we look at a finer parameter, this interpulse interval, the time between two pulses in the female. We quantify female IPI when the female is singing by herself versus when she's singing near or with a partner. So what we see is that there is a significant reduction in IPI values when she's singing with her partner, and when we remove the arista, this disappears. So this represents modulation of song pattern based on auditory cues. Finally, we return to the response time curves to see what happens when we remove the arista. So this is once again that wild-type female response curve, and when we remove the arista, it looks identical. So therefore, hearing is not required for female coordination with her male partner. However, when we look at the male response times, when we remove the male arista, rendering him deaf, the latency in his response time disappears, becoming random. So this would suggest that the male is listening to the female in order to acoustically coordinate with her. So I hope I've convinced you that these species perform an acoustic duet because I've showed you that both songs are important for the success of the courtship, that the songs are coordinated in time, and that hearing is involved in the patterning song, but in just different ways for males and females. But what we really want to know is where the female song is coming from, and we want to know this in terms of the neural circuitry. So in order to get a crude sense of this, we performed a series of manipulations on the female by removing or blocking various sensory organs, because we don't have any genetic tools in this species at all. So we waxed over the genitalia, amputated the tarsi, the arista, or the whole antenna itself, or placed courting flies in the dark to remove visual cues. We performed all combinations and permutations of these manipulations, and quantified female pulse rate. And so this whole data set can basically be boiled down to a decision tree. And what we find is that the genitalia is the most important sensation in order to produce female song. And this makes sense in a way because if you looked closely at the video, the male actually remains in contact with the female genitalia the entire time through touching her with his front forelegs and his proboscis. And then the next most significant sensation is in the arista. And the combination of these two senses results in different mean pulse rates. In order to confirm the importance of inputs from the genitalia in song production, we paired a wild-type male with a decapitated female, no head. And in this video, you will witness our extremely surprising and very new result, this was just last week, that a headless female has the capability to produce song upon male contact with her genitalia. Female, male. So we're further exploring this manipulation in the coming weeks. to see if this is actually a means of female coordination with the male. Finally, we wanted to turn to evolution to provide clues about the origins of female song. In the unilateral communication model of Drosophila and Melanogaster, the males produce two types of song, a pulse and a hum song, and the females are silent. Drosophila virilis diverged from Melanogaster about 60 million years ago. And so from our preliminary data, we showed that a subset of the males also produce two types of songs. The primary song is typically found in courtship type situations, whereas the secondary song is more of an aggressive song, but it is produced in the courtship just rarely. And then almost all of the females of this group produce courtship song as well. We analyzed the relationships between these songs on the basis of their song parameters. When first looking at interpulse interval, the time between pulses, what we see is that the strongest correlation is between female song and male secondary song. Similarly, male secondary song and female song have the strongest correlation for fundamental pulse frequency as well. Now this is just preliminary data, but female song seems to be co-evolving with male secondary song, so we hypothesize that virilis females have song circuits analogous to that of the male, rather than something new and different. This hypothesis is the drive for our future neurogenetic experiments. In Melanogaster, a neuron-specific transcription factor called fructose has been proven to be the master regulator of male behaviors. Because of this sex-specifically spliced exon here, this protein is present in males and absent in females. We show that there are similar expression patterns of this protein in virilis as well. It's conserved. So you can see here there are certain clusters in Melanogaster pointed out, and they're also present in virilis. And in female virilis, there's nothing expressed. So because of this conservation, we're using what we learned from Melanogaster and attempting to antagonize this protein in virilis using the CRISPR-Cas9 system in order to locate the female and just general song circuitry of this species. With this information, we can begin to build a model for the acoustic basis of female courtship song production. So first, the male will initiate the courtship by touching and singing to the female, and this is what turns the female circuit on. Then the female has to decide if she's receptive. If she's not, she'll sing minimal song, which leads to reduced copulation. But if she is, she'll sing a normal song. Then she has to decide if she's hearing male acoustic cues. If she's not, she sings at this elevated female pulse rate, which leads to reduced copulation. But if she can hear him, then she sings at a suppressed rate. Then in a more dynamic decision-making process, she has to decide if she's currently singing with the male in a duet. And if she's not, she sings at a default IPI rate. But if she is, she decreases her IPI, which eventually leads to a successful copulation. So I'd like to thank you for your time, and I'd also like to say that none of this data is published, so I'd really appreciate to bilaterally communicate with you after the talk to get some feedback on my experiments. And just to thank my lab, the Murthy lab, my thesis committee for all their help, and our collaborators on song segmentation analysis. Thank you.
You showed that, or you hypothesized that potentially a deaf female, if she's not hearing a male, will increase her singing. But could it also be that she's not hearing herself, and she might not have that feedback to check in with herself how much she's singing?
Yeah, it's very possible. We haven't really thought of a way to get around this. We've done some playback assays to these guys, and the females don't really seem to respond to playback assay at all. That would be a way that we would want to go about experimenting on this. And they just don't seem to respond to playback at all. So that would be something we would love to test, but just haven't been able to. Yes?
Sure, the males who are deaf don't respond to when they're blind and deaf. They do. But did you try doing males that are just blind? So you said this is because they need an acoustic signal that only becomes a visual signal. But have you tried with just the visual signal?
Yes, so the control for that experiment was a blind male, also in the dark. Yes. Thank you for that. One more quick question.
Do they respond auditorily in sound to chemical cues?
Chemical cues. We have not tried that yet. I do not know. But not so far that our knowledge consistent that happens. All right. So our next speaker